Here's some background to know for thinking about DNA. Welcome to Critical Thinking Scan, where we look at how you can think about any faith-challenging message and arrive at a biblical, logical conclusion yourself. I'm Patricia Engler, and today we're going to begin a series on thinking about evolutionary arguments which are based on genetics, genes, and DNA. First, it's important to start with some background info on the science we'll be talking about. So let's cover those basics now, looking at what DNA is and how it works. As you probably know, DNA contains information to build and maintain all living things. The fact that living things contain information is a pretty good clue that life requires a designer, as such information comes only from intelligent sources. But you can learn more about that in the linked resources. The information is encoded in 4 letters called nucleotides, which are initialed A, T, G, and C. The nucleotides are arranged in specific sequences along a backbone of sugar and phosphate molecules to compose a single strand of DNA. DNA is made of 2 strands wound together, creating a double helix, which looks kind of like a twisted ladder. The ladder's rungs are made up of paired nucleotides, where A always pairs with T and G always pairs with C. Genes, which are like messages written from these nucleotide letters, code for proteins. They're the molecules that do all the work in the cell. Now, something cool about DNA is that one sequence of nucleotides can contain information for multiple genes and multiple proteins depending which sections of information are copied and spliced together. So a DNA sequence is like a sentence that contains multiple functional messages depending which letters and words you read and which way you read them. Non-coding sections of DNA, which don't contain genes, control how and when information from the coding regions gets assembled to make different messages. All the DNA in your cells gets packaged into separate chromosomes, kind of like the way that sentences in books are packaged into chapters, but much more complex. All the information in the chromosomes makes up your genome, like chapters make up a book. So that's how the information in your cells is organized. To make proteins using this information, an array of other protein molecules works together to bind a specific DNA sequence, unwind the DNA, and use one of the DNA strands as a template to produce messenger RNA, also called mRNA. mRNA is also made up of nucleotide letters, but uses U instead of T. Individual mRNA nucleotides align with the appropriate DNA nucleotide to make an mRNA sequence, a strand with a complementary sequence to the DNA strand. Each set of 3 nucleotides along an mRNA strand is called a codon, and each codon codes for a specific amino acid, which are the building blocks of proteins. Translating mRNA's information into proteins requires other molecules still, called tRNAs. Each tRNA molecule contains an amino acid and 3 nucleotides, which are complementary to the nucleotides in specific mRNA codons. That's how codons can instruct the right tRNA to add the right amino acid in the right order to make a peptide chain which will become a protein. The order of amino acids in the chain helps determine the final protein structure and function, which is why having the right nucleotide sequence in the original DNA strand is so important. If the information in the DNA is being used to make mRNA, we say the DNA is being transcribed. If the mRNA which is transcribed from a gene is being used to make a protein, we say the gene is being expressed. That's a lot of info we just covered, and you can take entire classes which still hardly scratch the surface of how amazing and complex the interdependent systems of genes and proteins in our cells really are. You can learn more about all of this in the linked resources, but these basics will come in handy for thinking through evolutionary arguments about DNA, which we'll begin diving into next time. Meanwhile, for more on how to think critically about faith-challenging messages, you can access my other CT scan videos packed with tactics, tips, and tools that helped me as a Christian student at secular university. Thank you for watching. Hey, it's Patricia here. Just wanting to let you know that if you like these videos, a free, easy way to help Answers in Genesis Canada create more content and equip more people to defend their faith is to hit that like button, subscribe to our channel, click the bell, and of course, share these resources. That lets the social media algorithms help these videos reach more people who can benefit from them, saving us advertising expenses while promoting biblical authority. Thank you so much.